Hi, this lecture is about patterns of inheritance. This is how we inherit traits on our chromosomes. Those traits follow different patterns, and you'll understand what that means a little bit better when we go through some examples. The first person to really study this topic and publish was an Austrian monk named Gregor Mendel, so it's sometimes also called Mendelian genetics. Just a quick reminder that you have 23 pairs of chromosomes, and this is a colorized picture of those 23 pairs of chromosomes. Remember that one of each came from the egg that made you, and one of each came from the sperm that made you. And when those came together, they formed a zygote, that first cell that divided many times to make all you. Every cell of your body came from that original zygote. That 23rd pair of chromosomes are called the sex chromosomes. And depending on if you're genetically male or genetically female, those are going to be different. But those first 22, chromosomes 1 through 22, are called the autosomes. And when we refer to traits that are inherited on those first 22 chromosomes, we call that autosomal inheritance. So when we use the term autosomal, we're referring to those autosomes, the first 22 chromosomes. And males and females have those same 22. The 23rd pair, called the sex chromosomes, traits are going to be inherited a little differently there, because if you're genetically female, you have 2 X's, and if you're genetically male, you just have 1 X, but you also have a Y. Genetic females do not have that Y. So traits that are inherited on the Y chromosome are not going to be present in an individual who is XX. Also, if you have only 1 X chromosome and you have a bad gene on that chromosome, you don't have another copy of that gene to make that protein correctly. And that's going to be a big deal when we talk about traits that are inherited on the X chromosome. So when we're going through these patterns of inheritance, a couple of really important things right off that are important to consider. One is, where is that trait inherited? Is it autosomal, or is it on the sex chromosomes? That's going to be important. Then what pattern it follows is going to be important as well. There's going to be a lot of vocabulary associated with this lecture, just to warn you right from the beginning. And you will probably want to take really good notes, because these terms are very important, and we will use them again for other topics this semester. Let's start with just a quick reminder that you do have 2 of each chromosome. So let's randomly say that this is chromosome number 3. And somehow I chose the crayon there. So if it's looking fuzzy, that's not your screen. That is because I accidentally chose that. And let's now say that there is a trait that is on this chromosome number 3, and this is the one you got from dad. In other words, this is the one that you got from the sperm that made you. And this chromosome number 3 is from mom. In other words, this is from the egg that made you. That's how you got 2 of each chromosome. If there's a specific trait on this chromosome number 3 from dad, then your chromosome number 3 from mom has that same trait in that same location. But since mom and dad are not identical twins, the DNA sequence for that trait is different from both of those parents. So even though it's a trait that codes for the same protein in that same location, the version that you got from mom and the version that you got from dad, different DNA sequence. Nevertheless, we know that you can inherit the same version of that gene. In other words, the same allele from each parent, or you can inherit 2 different versions. Let's just start from the very beginning with some pretend traits. Let's assume that these are rabbits and not humans. And this is chromosome number 3 in rabbits. And this rabbit on chromosome number 3, let's pretend that this is the fur color gene. And let's assume that in these rabbits, there is gray fur or there is white fur. And those are the two possibilities. Gray fur and white fur. Those, remember, are called alleles, different versions of the same gene. This rabbit might have inherited gray from mom and gray from dad. Or maybe they inherited white from mom and white from dad. Or maybe they inherited one of each, gray from one parent and white from the other. Those are the possibilities. If these are the only two versions of this gene, in other words, if there are only two alleles for this trait, then they either got two the same or one of each. And there are some important terms associated with that. So let's draw out those three possibilities. This is going to be possibility number 1, number 2, and number 3. And those are meant to all be the same size, even though I drew those little sausage-shaped chromosomes, different sizes. Those are meant to be the same. So this is the same chromosome, chromosome number 3, and here's that fur color gene. And it doesn't really matter in this situation if these are from mom or from dad. So just remember, this rabbit has 2 copies of that chromosome. And let's say that this is one of the autosomes, because it's chromosome number 3. And chromosome number 3 in rabbits is an autosome. It's not a sex chromosome. And there are 2 different alleles for fur color. And let's say possibility number 1, this rabbit got gray from one parent and gray from the other parent. So they only have gray alleles, 2 the same. In this situation, this rabbit got white and white. And now this rabbit got one of each, gray and white. There are terms associated with that, so you know what 2 alleles that rabbit inherited. If a rabbit or any individual has 2 of the same version of a gene, they are called homozygous for that trait. This term homo means same. And zygous is referring to the zygote, or that very first cell that formed when egg and sperm came together. But in this case, it's not referring to the whole zygote. It's just saying for that gene, you have 2 that are the same. So that first individual would be homozygous. This individual would also be homozygous, meaning 2 alleles are the same. In other words, 2 of the same allele for that trait. This third individual, though, both those versions are different, and this individual is called heterozygous for this trait. Hetero means different. And that means this individual has 2 different alleles for that trait. Now we need a term to differentiate between having 2 gray versus 2 white. And in order to understand that term, we need to take this story a little bit further. In all of these patterns of inheritance that we talk about, these heterozygous individuals are going to be incredibly important, because how that gene gets expressed in the heterozygous individual is going to tell you what pattern of inheritance you're looking at. If you get 2 different versions of that gene, how that gets expressed is going to be really significant in telling you what pattern of inheritance that trait follows. In this pretend version of fur color, if a rabbit has one gray fur allele and one white, in other words, they're heterozygous for fur color, and if that individual ends up being gray fur, what that tells us is gray is considered to be dominant. So gray fur in a heterozygous individual for fur color tells us that gray is dominant over white. So when you have one of each, gray is the one getting expressed. That means white is what's called recessive. So gray is dominant and white is recessive. Let's define what dominant and recessive mean in this example. What they mean is a dominant allele is the one that is expressed in a heterozygous individual. Okay, whereas the recessive allele is the one that is not expressed. In a heterozygous individual. So now that we've established that in this example, gray is the dominant allele and white is the recessive allele, now we can go back to our homozygous individuals and we can tell whether that individual is homozygous dominant, meaning they have two of the dominant allele, or homozygous recessive, meaning they have two of the recessive allele. So let's go back here. This individual, gray and gray, are the two versions of that gene they have. So they're homozygous, and we would now say that they are homozygous dominant, meaning they have two of the dominant allele. And this individual that has white and white, they are going to be what's called homozygous recessive. They have two of the recessive allele. So that's how we differentiate between those two. The only way we know which trait is dominant and which one is recessive is, again, looking at the heterozygous individual. The trait that gets expressed in a heterozygous individual is the one that's dominant. Two more important terms right now that go along with this whole dominant, recessive, homozygous, heterozygous. And that is genotype is the two alleles an individual has for that trait. And then the phenotype is really how that genotype gets expressed. It's the physical expression of the genotype. Going back to the rabbit fur color as our example. Sorry. That first rabbit was gray and gray. That was the genotype. And the phenotype is going to be how that genotype is expressed. In other words, What fur color does a rabbit have if they got a gray allele from one parent and a gray allele from another parent? What fur color would they have? Gray fur. They don't have any other option. They got gray and gray, so they're gray. The rabbits that got white fur gene from each parent, you can probably guess, they got white from one parent, white from the other. That's their only option. They have white fur. That's their phenotype. Here's where it gets significant. Again, the heterozygous individual. If a rabbit gets gray from one parent and white from the other, and these are autosomes, so it doesn't matter whether the gray came from mom or dad or whether the white came from mom or dad. We've already said that gray is dominant over white, so the phenotype of these individuals is going to be gray fur. In order to predict what offspring are going to look like based on the genotype of parents, scientists came up with a method for doing what are called crosses between individuals, looking at what the possible eggs are and the possible sperm when individuals mate. And they do that using a square called a Punnett square. And I'm going to show you that in a minute. But in order to do that, we have to represent the genotype with a letter. And that letter is going to be used in our cross. And typically, what happens is the dominant allele is represented with a capital letter, and then the recessive is represented by a lowercase of that same exact letter. And I'm going to show you how that works. So gray, we've already determined is dominant. We know that because in the heterozygous individual, gray is the one that gets expressed. So we're going to represent gray fur allele with a capital G. So the genotype of this first individual is big G, big G. In other words, 2 gray alleles. We're now going to represent white as a lowercase g. Why are we not using a W? Because if we use a W, that doesn't tell us anything about which one's dominant and which one's recessive. If we just had G and W, it wouldn't tell us anything about which one gets expressed in the heterozygous individual. So white is going to be lowercase g. That means this third individual, the heterozygous individual, is big G, little g. And let's say that we have a rabbit that is heterozygous. So we have a heterozygous mom rabbit, and we have a dad that's homozygous recessive. If dad is homozygous recessive, that means dad is little g, little g. 2 of the same, that's the homozygous part. And recessive tells you that it's 2 of the white gene. Mom is heterozygous, that means she's big G, little g, 1 of each. This is called a cross. We're crossing a heterozygous mom rabbit with a homozygous recessive dad rabbit. And we're just looking at one trait. So this is a cross that's just looking at one trait. When you're just looking at one trait in the square, it's actually called a monohybrid cross. It means you're just looking at one trait. Along the top, it doesn't matter if you do eggs on the top or eggs on the side, but we're going to do eggs, possible eggs from mom here on the top, and we're going to do possible sperm from dad on the side here. Remember that during meiosis, wow, meiosis is significant here. In meiosis, each of these rabbits are going to make gametes. And During meiosis 1, this pair of chromosomes is going to get split up. And this little g is going to go to 50% of the eggs, and the big G is going to go to 50% of the eggs. So that means our possible eggs are 50% are going to get big G and 50% are going to get little g. In Dad, he's homozygous recessive. The only trait he has to give to sperm are little g. So 50% get this little g and 50% get the other little g during meiosis 1. So sperm, little g, little g. Now what happens in these squares are all of the fertilizations. This egg comes together with this sperm, and that gives us big G, little g for this offspring. Then this big G crosses with this little g. This little g crosses with this little g, or they come together to make this individual. And then the same here, this one comes together with this one. So it's all the possible fertilizations. This shows you what all of the possible outcomes are for offspring. Each one of these squares represents on average 25% of the offspring. And the way this would get reported is we would say these two together equal 50%. So we would say 50% of the offspring are big G, little g, and 50% are little g, little g. That represents the possible genotypes of our offspring. We now need to look at the phenotypes of the offspring. How does this get expressed? Remember that big G, little g, heterozygous. Those are going to be gray fur. And little g, homozygous recessive, that's going to be white fur. So on average, 50% of the offspring are going to be gray and 50% are going to be white. Just so happens they end up looking just like the parents in this cross. So this square that helped us figure that out is called a Punnett square. I wrote these really close together. It's 2 N's and 2 T's. Obviously a person's last name, probably the person who figured this out. So a Punnett square. Sorry, I'm going to write that again. Looks like that. Punnett square is how we figured this out. This cross in which one allele is completely dominant over the other allele is our first pattern of inheritance. And it's called complete dominance. In this story about fur color in rabbits, gray was completely dominant. Gray was completely dominant over white. In complete dominance, one allele is completely dominant over another allele. And what that means is in a heterozygous individual, that heterozygous individual, has the dominant phenotype. In other words, whatever that dominant trait is, that's what the heterozygous individual looks like. In other words, in this example, big G, little g gives you gray fur, which means gray is completely dominant over white. To be more exact, gray fur is completely dominant over white fur. This is the way that many, many traits in humans are inherited, is through this first pattern of inheritance called complete dominance. And this is especially significant when we're talking about something more serious than fur color. when we're actually talking about disease traits, genetic disorders. A lot of genetic disorders follow this pattern of complete dominance. Let's now take it a step further and talk about complete dominance with respect to genetic disorders. The way that these are inherited through complete dominance, I'm going to use cystic fibrosis as our example. Cystic fibrosis follows this pattern of complete dominance. And in this pattern, big C is normal. And it's hard to, I'm doing this really exaggerated because this letter C is really hard to distinguish between the capital and the lowercase. Little c is going to be cystic fibrosis. Let's talk for a minute about what this gene that's affected by cystic fibrosis codes for. In your cell membranes, you have proteins that allow salts to move in and out of the cell. It's a transmembrane protein. And in cystic fibrosis, the DNA code for that specific protein is incorrect, and it codes for a misshapen protein, and salts cannot move in and out of the cell. Salts accumulate, and in response, the body forms mucus, especially around the lung cells. This mucus damages the lungs, the cells of the lungs, and other cells in the body, and over time ends up resulting in the death of this person. It's a very, very tragic disorder to inherit because there is no cure currently for cystic fibrosis. We have done things to dramatically increase the lifespan of people with cystic fibrosis. Average lifespan used to be around 12, and now that average lifespan is even up in the 30s, which is amazing. But sadly, these people mostly all die. If you have 2 copies of this gene, which we all do, and you have one bad gene, let's say that this one is little c. So long as you got one good copy of that gene, you are making enough normal protein to not have full-blown symptoms of that disease. This is going to be the bad copy of the gene. So you're not making normal protein. But so long as you have one normal copy of that gene, you often don't have that disorder. When that's the case, then that means that trait is inherited through the pattern of complete dominance. It's also important to note that that only is the case if it is autosomal. So it has to be an autosomal disorder, meaning it's inherited on chromosomes number 1 through 22, not on the 23rd pair of chromosomes. And in this case, the disease is recessive and normal is dominant. So if normal is dominant and the genetic disorder is recessive, then what we call this disorder is we say that this is an autosomal recessive disorder. Autosomal tells you it's inherited on chromosomes 1 through 22. Recessive tells you it's inherited on the recessive version of that allele. That means normal is dominant over abnormal. In other words, so long as you have at least one normal copy of that gene, you're going to make enough normal protein to not have full-blown disease. This is the way that many, many disorders in humans are inherited. They are autosomal recessive disorders. This is not always how diseases are inherited. So this is only if something is called autosomal recessive, that this is how it's inherited. And in that case, you have three possibilities. To keep it a little straighter on these crosses, I'm going to now for the crosses use a condition called albinism, where a person is born without melanin in their skin. So their skin is really, really light in color. And albinism is autosomal recessive. So we would represent it with lowercase a for the genotype. Normal pigment in the skin is going to be a capital A. This tells you that normal is dominant over the disorder. So three possibilities. A person could get two normal, one of each. Oops. Or two of the disorder alleles. This person would be homozygous dominant, heterozygous, homozygous recessive. The only way you can have an autosomal recessive disorder is if you're homozygous recessive. Because if you get even one good copy of that gene, you're going to make enough normal protein that you don't have the disorder. Let's do a cross, a Punnett square, a monohybrid cross with albinism. Let's say that mom is normal pigment and she is homozygous dominant. Oops. That means mom is big A, big A. And let's say that dad is heterozygous. In other words, he's big A, little a. Let's do this cross. We'll do mom's possible gametes on the top. Mom only has big A to give, so 50% of the eggs are going to get this big A and 50% are going to get this one. So big A, big A. Dad, 50% of the sperm get big A and 50% get the little a. So let's do our cross. Big A, big A, big A, big A, big A, little a, big A, little a. So this, and then this, and this. So 50% of the offspring have normal skin pigment and 50% are heterozygous and also have normal skin pigment. So for the phenotype, it's going to be 100% normal. But for the genotype, 50% are going to be homozygous dominant and 50% are going to be heterozygous. If we're talking about a gene that causes a genetic disorder, we call those heterozygous individuals something different. And that is, we call them carriers. Carriers are heterozygous. They carry the disease trait and they have the ability to pass that on to their offspring. but they don't have the disease or disorder because they are heterozygous. This normal gene is completely dominant. So they don't have the disease, but they're called carriers because they carry that in their genotype. And look what happens if two carriers mate. So two carriers mate. That means big A times big A little a. And let's do the Punnett square for that. So on average, by the way, these averages assume a lot of offspring. So if this was an insect laying thousands of eggs, this would be the ratios. But if you're a human having babies, every single time your offspring have a 50% chance of getting this gene in the egg and this gene in the sperm. So these odds really aren't true odds if you're only having a few offspring. This would be only if you're having thousands of offspring. And that's how you can have in families multiple children having a genetic disorder when the parents were both carriers. The odds of that are slim, but it can happen, and it does happen. So looking at the percentages here, 25% are big A, which is normal. 50%, remember each square represents 25%. So those two together are going to be 50% are going to be what we call carriers. And it's important to differentiate between the carriers because the normal individuals don't even have the bad gene in their DNA, but the carriers do. So even though they have normal skin pigment, we call them carriers because it's a genetic disorder and they are heterozygous, which means they could pass that on to their offspring. And here we go. These are the individuals that have the disorder. So 25% on average, little a, and these individuals have albinism, which is the genetic disorder that we were tracking with this cross. And again, every time mom makes eggs, 50% get the bad gene, and every time dad makes sperm, 50% get the bad gene. So it could be just by a horrible roll of the genetic dice that they have 3 offspring and all 3 of them end up having albinism or something worse. You know, there are genetic disorders that are deadly at a very early age, things like Tay-Sachs disease, where the baby inherits a genetic mutation that causes a key enzyme to not be present or not be produced correctly. It's misshapen. And that baby has fats that accumulate around the brain, and they go blind, deaf, they have multiple seizures per day, and they end up dying by age 3 or 4. Some horrible, horrible genetic disorders that are inherited this way. The only way the baby can have the disorder is if they have 2 bad versions of the gene. And that's because these are autosomal recessive disorders, meaning that normal is dominant. So if you are a carrier, you don't have the full-blown disease. You're making enough normal protein to not have symptoms of that disease. So these, again, are only in genetic disorders that are autosomal recessive. There are disorders that are what are called autosomal dominant, and we will talk about those as well. Autosomal dominant disorders are a little more confusing. In order to really understand autosomal dominant disorders, I need to just remind you about autosomal recessive disorders again. I'm going to continue to use big A, little a for a while, just because it's easy to differentiate those 2 letters. So remember that in autosomal recessive disorders that we just talked about, big A is normal. Big A, little a is what we call a carrier. And little a means they have the disorder. You have to have two of the recessive gene to have the disorder. In the world of autosomal dominant disorders, it's different. It's a different outcome. And that's because in autosomal dominant disorders, little a is normal and big A is the disorder. In other words, the disorder is dominant over normal. And in that case, this individual has the disorder and this individual has the disorder because this is the disease gene and this is normal and disease is dominant over normal. So that one gets a little more confusing and I'm going to give you examples of this. So normal is recessive. In this pattern of inheritance. So the only way an individual could be normal is to get two normal copies of the gene. The way you can tell the difference is if an individual is heterozygous and they don't have the disease, it's following this pattern. If they're heterozygous and they do have the disease or disorder, then it's following this pattern. So that becomes a little more confusing. In most cases, this combination is so bad that this is lethal and that baby never develops. So usually in these disorders, that doesn't even exist by the time that person would be reproducing. So that's a lethal disorder because it's too much of a bad thing. Remember, this is the bad gene and you've got two copies of the bad gene. That's really bad. If it's bad enough that having one of each, you have the disorder, then having two is really bad. Classic examples of this. One is Huntington's disease. Huntington's disease is a very sad degenerative disorder. It degenerates your central nervous system. And people usually start showing symptoms of this very young, like even in their 30s. And by then, unfortunately, most people have, had children because some people don't start showing symptoms till like their 40s or 50s, but it's still much earlier than most degenerative brain and central nervous system disorders. So these people have signs of dementia pretty early on, but they also lose coordination of their body. Huntington's disease is autosomal dominant. In other words, it's an autosomal dominant disorder. So in this case, I'm going to use H, since it's Huntington's disease. Big H is Huntington's disease. We know that it's the big H because it's autosomal dominant. That means the disease is carried on the dominant allele, and we use the capital letter for that. That means little h is normal. Three possibilities: big H, lethal. Too much of a bad thing. Heterozygous individuals have Huntington's. Because there's enough bad here, it's dominant, but the disease is dominant over normal. The only way an individual could be normal is if they had two of the little h. Let's say that an individual with Huntington's disease who doesn't know yet that they have it reproduces with someone who's normal, meaning they don't carry the gene for Huntington's disease. And in fact, they're not even called carriers because remember, carrier means you have the disease gene, but you don't have symptoms of the disease. These people have full-blown Huntington's disease if they are heterozygous. So Huntington's disease is going to be big H, little h. How do I know this? Because if they were big H, they would not be reproducing. That would have been lethal. Normal is little h. So this cross, draw our Punnett square. It's autosomal, so it doesn't matter which parent has Huntington's disease and which one is normal. It doesn't matter if it's mom or dad, so I'm just leaving that out of the story. So sadly, if you look at this, on average, 50% of the offspring are going to have Huntington's disease. And these 50% here are completely normal and they don't carry the disease allele in their DNA. So this would be 50% big H, little h, which is Huntington's, and 50% little h, which is normal. Let's do another cross with an autosomal dominant disorder. Achondroplasia, sometimes called dwarfism, is an autosomal dominant disorder. That means disorder is dominant and normal is recessive. Big D is lethal. So that means people with dwarfism are big D, little d. And little d is normal. So if I said that two parents have dwarfism, you would know that they are both big D, little d. Two heterozygous individuals this is what our cross looks like. So remember, each square is 25%. So 25% of the crosses are going to be lethal. 50% are these two, dwarfism. But here's what's crazy, 25% little d, normal. So these individuals, both parents had dwarfism. Not only do they not have dwarfism, but they can't even pass it on to their offspring because they don't even have that gene in their DNA. And that's because mom and dad were both heterozygous. So if heterozygous individuals have the disorder, you know that it's autosomal dominant. There's no way for it to be autosomal recessive because in that case, these heterozygous individuals would not have the disease or disorder. So again, quick reminder, if it's autosomal recessive disorder, we're looking at the heterozygous individuals to tell us big A, little a is called a carrier, no disease. In autosomal dominant, that's not the case. In an autosomal dominant, the disease is dominant over normal. So in an autosomal dominant disorder, big A, little a has the disease or disorder. So again, looking at the heterozygous individual is going to be significant to know what pattern of inheritance you're looking at. Now we're going to look at something called incomplete dominance. We've talked about complete dominance. Now we're looking at incomplete dominance. So everything we've talked about so far is that pattern that's called complete dominance. Let's say that We're looking at a plant that is a flowering plant, and big R gives red flowers, and little r gives white flowers. If this was complete dominance, heterozygous individuals would most likely be red. But what if I told you that big R gave pink flowers? In this case, the heterozygous individuals end up with what's called an intermediate phenotype, something that's in between. In other words, both of those genes are having an influence on the flower color. Red is having an influence and white is having an influence. It's almost like a blending of the two. So you end up with an intermediate phenotype. So heterozygous individuals have an intermediate phenotype. A couple of examples of this in humans. Straight hair times curly hair gives you wavy hair. The heterozygous individual has an intermediate phenotype. LDL receptors on your cells. LDL, this is the bad cholesterol. I'm going to use big L as my gene. We're going to say big L. Lots of LDL receptors on your cells. These are people who can eat French fries every day and not get high cholesterol. We don't like these people. Just kidding. It's just not fair. Little l, no LDL receptors. These people get high cholesterol from an early age. It's genetic. They don't have enough LDL receptors to break down cholesterol if they have a poor diet. If you're intermediate, it's because you're heterozygous, and this is an intermediate number of LDL receptors. So if you eat healthy, you're not going to have high cholesterol. If you eat unhealthy, you'll probably have high cholesterol. So again, incomplete dominance, heterozygous individual ends up with an intermediate phenotype, somewhere between the dominant and the recessive. Not as common in humans. Incomplete dominance is that pattern. So we've talked about complete dominance, incomplete dominance. This next one is a little more confusing. It's called codominance. Oops. And the best example of a trait that follows this pattern of codominance is ABO blood type in humans. We're going to do a blood typing lab. It's the very last lab of the semester that we do in week 13. And in human blood type, there are two major markers on your red blood cells that can be inherited. One is the ABO blood type marker, and the other is what we call the RH marker. So we're not talking about whether you're positive or negative. So if you have O positive blood or O negative blood, we're not talking about the positive and the negative. That RH factor is inherited by a pattern called complete dominance. I'll talk about that a little more in a minute. We're talking about, are you type A, B, AB, or O? Oops, sorry, I turned my iPad on accident. So this is how it goes. There are 3 possible alleles for human blood type. And they look like this. They're going to look a little bit different than everything we've looked at before. This is the type A allele. This is the type B allele. And this little I is the type O allele. Again, I'm going to define codominance for you after I give the example because it will make more sense then. So let's look at the possible genotypes and the possible phenotypes for ABO blood type in humans. And when I tell you this story, I'm also going to explain what that big I and that little I mean. A person can get 2 of the type A allele, one from mom, one from dad. We would say that this individual is homozygous, meaning they have 2 of the same allele. They are homozygous type A, but their outcome is that they have type A blood. What that gene codes for is a specific marker on your red blood cells, only on your red blood cells. And if this is your red blood cell, I'm going to draw a pretend marker shape. Your red blood cells would have this type A marker. Even just having one gene that says make the type A marker is going to give you the type A marker. This person has 2 of them, so they definitely have type A markers on their red blood cells. Again, I'm going to tell you why that's a capital letter I in a second. The other way that you can be type A is to get 1 type A allele and 1 type O allele, because type A is completely dominant over type O. So this individual would be heterozygous for type A blood. And in this case, this is saying make type A markers, and this is saying make no markers. Because the type O allele says don't make any markers, A, B, or otherwise, on the red blood cells. Now, again, this doesn't have, the RH marker is a separate marker. This is just the ABO blood type marker. So if you've got one gene saying make type A markers and one saying make no markers, you're going to make some type A markers and you're going to have type A blood. The same thing goes for B, except that it's a different marker. So you can be homozygous for type B blood. So this person is type B blood. The other way they could be type B is to have one gene that says make type B markers and one that says make no markers. So type B markers are going to be a different shape. So in this example, let's give them a different shape. So if you even have one gene saying make type B markers, you're going to make type B markers, and they have a different shape. The only way that you would have no A, B, or O, A or B markers on your red blood cells is to get 2 of the type O. This is the only way you can be type O. And again, that little I says, this is type O, no A or B markers. What if you get an A gene and a B gene? Say you're this. In other words, one of those genes, this one, is saying make a type A marker, and this one is saying make a type B marker. You're making both because you have one of each kind of gene. And in that case, you're going to have type AB blood. Okay, so what is codominance? In codominance, two alleles are not dominant over each other. They both get expressed. They can be dominant over a third, but they're not dominant over each other. They're what's called codominant. So if you get one of each, they're both going to be expressed. And it makes sense if you think about what that gene is coding for. O says don't make any markers. So it's recessive, right? Because if you have another gene saying make markers, you're going to make markers. These heterozygous individuals, you can see that B is dominant over O and A is dominant over O, but they're not dominant over each other. So if you have an A and a B, they are codominant. So in codominance, and sometimes codominance is hyphenated and sometimes it's not. It depends on the textbook you look at. Either way is acceptable. Codominance, two alleles are not dominant over each other. Both get expressed. Okay, why the big I and little I? Big I is referring to immunoglobulins, and it's because if you have a marker that your immune system doesn't recognize as self, it's recognized as foreign, and you are going to produce antibodies against that blood type. So if you have just type A markers and your type A blood, if this is your genotype, you recognize type B markers as foreign, and you produce antibodies against type B markers. And if you ever have got any blood with a type B marker on it put into your body, those Y-shaped proteins, the antibodies, so this is an antibody, it would bind to that red blood cell with the type B markers, and it would cause your blood to clump. And it's fatal very quickly. Same thing would happen in an individual that was type B if they got a type A marker put into them. It's only what your body recognizes as self. So type AB, they can receive type A blood because they recognize that as self. They can receive type B blood. They recognize that as self. Everybody can get type O blood because it doesn't have any markers to make anyone's immune system angry. So if you're type O, you can't get A or B. You recognize both of those as being foreign, and you're going to cause type A to clump and type B. So if you're type O, you can only get type O blood. Let's look at that chart for a minute of who can get what. So let's go right here. And this is kind of a logic problem. Blood type. Can donate to which blood types and can receive from which blood types. So let's go type A. So if you're type A blood, it means you have type A markers on your red blood cells. Who recognizes you as self? Others with type A. And also type AB. Type AB has type A markers, so they recognize as self. Who can type A receive from? Obviously type A. If you're type A, can you get type AB blood? No, you cannot. You would recognize that B marker as being foreign and clump that blood. But who else could you receive from if you're type A? Which blood type has no markers that would make your immune system mad? It's the type O. Everyone can get type O. Similar story for type B blood. You can donate to B and AB, but you can only receive from type B and type O. You couldn't receive from AB because the A marker would cause you to produce antibodies and clump that blood. If you're AB, guess what? The B part makes A mad, the A part makes B mad. They both make O mad. So the only person you can donate to if you're type AB is to type AB. That's it. But you're lucky because you can receive all blood types because you recognize them all as being self. You're called the universal recipient. You can get anyone's blood. Your type O, no ABO marker. No, I'm sorry, I call them ABO markers, but no A markers, no B markers. You can donate to all. You are the universal donor, but you can only receive from O because any A or B markers are going to be perceived as foreign. This story I've just told you does not include RH. RH is the positive and negative part. If you're RH positive, it means you have the RH marker on your blood cells. If you're RH negative, it means you don't. So if you're positive and positive, then you're RH positive. If you're positive and negative, this gene says make the markers, this says don't. You're going to make the markers, so you're going to be RH positive. The only way you can be RH negative is if you got an RH negative gene from both parents. This is a different marker. It's called RH because it was discovered in rhesus monkeys. This is the marker that if you have a baby with a different RH factor than you could have problems because you produce antibodies against that. And then the next baby you could have problems with. They give you a drug called Rogam to cause you to not make antibodies against that baby in the future. But that's a whole different topic. But what I want to express to you is that this is a different pattern of inheritance. It's complete dominance. And in this story, positive is dominant, negative is recessive. And the way we know that is because the heterozygous individual, this individual is RH positive. So that's how we know that positive is completely dominant. So the positive negative is a separate thing. So if you're A positive, it means you have the type A marker and you have the RH marker. Okay, if you're O negative, it means no A or B markers and no RH markers. So you're the true universal donor if you're O negative. If you're O positive, you can donate only to A positive, B positive, AB positive. So the RH factor is a consideration when donating blood. The true universal donor who can give blood to everyone is O negative. Type O is the most common human blood type. So at some point in human evolution, there must have been a survival advantage to not having an A or a B marker on your red blood cells. And that's interesting because if we go back here, remember that this is homozygous recessive. Usually recessive traits are less common in the population, but in this case, type O with no markers, no A or B markers must have offered a survival advantage. So maybe there was some type of parasite that attacked red blood cells and it only attacked those that had A or B markers. Hard to say. There are many parasites that attack red blood cells, including malaria, which is still a major killer on the globe. But malaria doesn't discern. Okay, malaria attacks all blood types equally, but maybe at some point in human history, there was something that didn't attack type O. And in fact, it's interesting because when coronavirus, COVID-19 version of coronavirus first started spreading, there was a lot of hype about if you had type O blood, you were less likely to get it. That we know now that that's not true, but there was some talk of that early on. There hasn't been a study that's confirmed that. Let's just say that at this point. Maybe at some point in the future, they might decide that there's a correlation there, but right now there's no proven correlation between your blood type and how prone you are to getting COVID-19 infection. Everything we've covered now, those are all the autosomal patterns of inheritance. So we had autosomal recessive and autosomal dominant disorders, and those all covered, complete dominance. So complete dominance was the pattern, and that included autosomal recessive disorders and autosomal dominant disorders. And there was incomplete dominance, where the heterozygous individual had an intermediate phenotype. That's where red and white flowers made pink. And then there was codominance, where if you have both alleles, they both get expressed. So type AB blood, if you have an A and a B, those both get expressed. If you have O, that's recessive, but A is not dominant over B and B is not dominant over A. They're both dominant over O. And that's because if you think about what they code for, it makes sense. Those were all autosomal. Now we're going to talk about that 23rd pair of chromosomes, and we're only going to talk about X inheritance, not Y. So we're going to talk about what's called X-linked inheritance. If a disorder is carried on the X chromosome, it's inherited differently in women than it is in men, different in males versus females. And again, this is looking at, are you genetically male? Are you genetically female? And do you have the normal number of X and Y chromosomes? There are going to be 5 possible outcomes. I'm going to use red-green color blindness as my first example of an X-linked trait. And the way I'm going to show an X that has the colorblind gene is I'm going to do a superscript C. This is going to be colorblind. It's not on the Y, so it's only on the X. Here are the possibles. This would be a normal female, meaning no colorblind gene. Both the X's are normal. They don't have a bad gene for colorblindness. This is going to be a carrier female, meaning that one of the X's has a bad gene and the other one doesn't. The only way these females could have color blindness is if both of their X chromosomes have the bad gene. So this would be a colorblind female. And I'm just going to draw a bad gene on that one, bad gene on that one. If you have one good working copy of the gene, you're making enough normal protein to not have the disorder. You're a carrier female. So in this carrier example, this normal X is making enough normal protein that this individual does not have the disease. Males are a different story. Males are XY, females are XX. So, oops, sorry, wrong one. This is going to be a normal male, meaning no colorblind gene. This male has color blindness. Why? He has a bad copy of the gene on his X chromosome, and guess what? He does not have another X. So he doesn't have another opportunity to make a normal version of that protein. He only has the bad gene. He only has one X. And so this is going to be colorblind male. There are no male carriers, because carrier means you carry the bad gene, but you don't have the disease. There are no male carriers because even just one bad copy of that gene, you don't have another X to balance that the way this person did. That's why more males have X-linked disorders. Okay, there are more males with color blindness. There are more males with hemophilia. There are a lot of genes on the X chromosome, and none of them have to do with gender. Okay, the X chromosome, we all have one. You have to have an X to be alive. You don't have to have a Y to be alive, but you have to have an X to be alive. So that X chromosome codes for a lot of things that have nothing to do with gender. If a male has a bad copy of a gene on that X, he does not have another X to balance that, and therefore he's going to have the disorder. These crosses look a little bit different because now the gender of the parent and the gender of the offspring matter, because the way that trait gets expressed in females is different than it is in males. So let's do a couple of crosses. Let's have a normal female mate with a colorblind male. And the way you draw this Punnett square isn't as important as reporting the outcomes correctly. And it's going to be different than the other Punnett square in reporting the outcomes, because now the gender of the offspring matters. So normal female, that means she's XX, no colorblind gene. The colorblind male, we're going to show that as a colorblind X gene and a Y that's just Y. It's not carried on the Y chromosome. So we'll do female here, XX, the male here, there's his colorblind X, and here's his Y. So 50% of his sperm are going to get the Y, and 50% are going to get the colorblind X during meiosis. So now let's do the crosses. X, XC, XY, XXC, XY. So this is interesting because now you'll see that when we report our offspring, everything up here, these are all our female offspring, and these are all our male offspring. So now each square represents something different. This is 50% of the females, and this is 50% of the females. So each of the squares now represent 50% of the females. This is 50% of the males. This is 50% of the males. It just so happens that they're the same in this cross. So we would say 100% of the female offspring are XXC, which is carrier females. So mom was not a carrier, but they all got that bad X from dad, so now they could pass that on to their offspring. And this is interesting. Dad had color blindness, but all the males are normal. No colorblind genes. Let's do another one. This time, let's do hemophilia. So hemophilia is an X-linked disorder. This is the only time that gender matters for the offspring and the parents. Don't suddenly now make everything X-linked. That's, I caution you, for some reason, once I introduce X-linked, people want to do X and Y for everything. Autosomal, you don't do X and Y. It's only when it's X-linked. Let's say that mom is a carrier and dad has hemophilia. So mom is a carrier. I'm going to use the letter H now instead of the letter C. So mom has that bad gene somewhere on her X chromosome, but she's a carrier, so that means she has a normal X also. Dad has hemophilia, so he's this genotype. Again, he has a bad gene somewhere on his X. So let's do this cross. I'm going to do dad's up here so you can see how this can look a little different. And we'll do mom's gametes over here. Now what's going to happen is our female offspring are going to be on one side and the males are going to be on the other. So it's going to be divided this way now. Instead of up here, it went this way. It's just to show you that it doesn't matter if you put dad's gametes on the top or on the side. The outcome is the same. So it shows you that now our females, those two cells look different. And the same for the males. This one's XHY and this one is XY. So this with this and then this with that. Remember that each one of these cells represents 50% of the females. So these are our females over here, and these are our males over here. And on our females, 50% of them are this, which is colorblind females. The other 50% of our females are this, which is carrier females. Okay, now our males, 50% are this, which is I'm sorry, it's not colorblind. We're doing hemophilia. These males have hemophilia. Remember, there are no carrier males. There is no other X to balance this. So here, these females, this is a normal X. She can make enough normal protein that she doesn't have hemophilia. But this male, he's got one bad X. He doesn't have another X to counter that. So this is a hemophilia male. And then guess what? Even though mom was a carrier and dad had hemophilia, 50% of the male offspring are going to be normal, no hemophilia gene. So that is only with X-linked inheritance. Don't start doing X and Y for every trait once you learn this one. X-linked inheritance, different from autosomal. There are also traits inherited on the Y chromosome, but we're only talking about traits that are inherited on the X. So there are other patterns of inheritance we haven't talked about in this class. There are times when it takes the combination of multiple genes to produce a trait, such as your eye color, your skin tone. We're not going to talk about those patterns. Those are a little more complex. You need to be able to do crosses with all of these patterns of inheritance. There are practice problems online as part of the study guide. So please do those practice problems. They represent one problem for each pattern of inheritance. Also, really pay attention to the questions that say, you know, you have this cross. What would it look like in codominance? What would it look like in incomplete? What would it look like in complete dominance? Also, don't forget, this is really important. I'm going to quickly scroll back up now because I want to show you something important. really understand the difference between autosomal disorders and autosomal dominant disorders. And again, look at the heterozygous individual. If it's a disease and the heterozygous individual has the disorder, in other words, big A, little a, sorry, I keep pulling the highlighter here, big A, little a, if they have the disorder, then you know it's autosomal dominant. If they're big A, little a, and they don't have the disorder, then you know it's autosomal recessive. So what that means is this big A is either normal or it's disease. If big A is normal, then you're looking at autosomal recessive. In order to get that disease, you would have to have 2 of the bad gene. But in autosomal dominant, this individual has the disease, because in autosomal dominant disorders, disease is dominant, normal is recessive. In autosomal recessive disorders, disease is recessive, normal is dominant. And remember, I drew a little chart for that way up here. And remember, the possibilities are this. So really make sure you understand that there are some questions on the study guide about that. And if you have questions about that, please come see me, and I can even give you more practice problems. But the practice problems on the study guide, if you can do those, then you'll be good on the exam. Remember, the exam is timed, and you need to actually have some scratch paper to draw out Punnett squares, because the answers are going to be multiple choice, but you're going to have to do these crosses on paper, and then I've got every possible outcome in the multiple choice answers. So you actually have to do the cross on scratch paper and then choose the correct answer. So you need to really be able to do a cross for each of these patterns of inheritance. Whew, I know this is a long lecture. Sorry about that. It's just a long topic. And again, I've only hit some of the patterns of inheritance. There are more than that we could have talked about. But thanks for your attention.